Hello, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. And today I am launching my new series on my course that I used to teach at the university for over 25 years called Plant Physiology. I want to share with you the experiences I had and the discoveries I made throughout my 25 years or more, my 30 plus years of research. I was fortunate that when I studied plant physiology, it was at the University of California, Davis, in the degree of plant physiology, which no longer exists. At the time I was there were over 120 plant physiologists on the campus. It was a special time for plant physiology. I want to share with you the things that I've learned through the study of plant physiology and my research. And I believe I have added extra things that are special to this plant physiology class. I want to transfer my knowledge to the future generations so that it is not lost. So bear with me as I go through a fairly technical class in plant physiology. It's an upper division graduate level course, and therefore the students should have some background in chemistry or biology or biochemistry. But in general, I will assume that you know very little and will refresh your memory on topics you may have already had. One of my goals of this course is to teach you how exciting and interesting plants are. They're much more complicated than you think, and I think you will know that by the end of this course. So let's get to it.
When I taught this course as a cross-listed course of biochemistry and biology, 410, 610, both an upper division and a graduate level course.
What is plant physiology? Plant physiology is the science of study of plant function. Plant physiology is the study of dynamic processes responsible for growth and development of plants.
Plant requirements are really quite simple. They need light for photosynthesis and the production of carbohydrates. They need CO2 for photosynthesis and the production of carbohydrates. For body structure, they need water for body structure and biochemical reactions. And they need minerals for body structure and biochemical reactions. Plants do not need vitamins as they are able to synthesize all that they need. In this course, we will study the transport of water, the transport of minerals, the transport of sugars. Then we will move on to photosynthesis and growth and development of the plants, genomics and gene expression, hormones, or also known as plant growth regulators. And then finally, we'll take all of this knowledge that we've learned over the entire course and integrate it in studying the environmental physiology of plants. That is, the response of plants to the environment. So in the first half of the course, we will be talking about transport. It will be somewhat mathematical because we are talking about transport processes. Then we will focus on energy, which includes photosynthesis and respiration, and how that all interacts into growth and development. And then the emerging field of genomics and gene expression over the last 20 years has caused an explosion of information and knowledge in the field of biology as a general rule, but also in plant physiology. Then we'll delve into at least 5 hormones, if not more, in which we know that they are major growth regulators.
At the end of each lecture video, there will be a quiz video. This is for you to try and to test yourself on your knowledge that you've gained. I find that having quizzes helps sink that information into your memory just a little bit better. So give it a try. I think you'll have fun at it. It's a multiple choice type quiz. And at the end of that quiz, you can look at another video which has the answers. These are actual quizzes that I gave in my plant physiology course during my 25-year career teaching plant physiology. Before we start talking about plants, let's briefly talk about the scientific method, as it is the foundation of modern science today. It's very important to understand the scientific method because it gives us the ability to predict the world around us in a more accurate way.
It's based upon a series of hypotheses that are followed by experiments which allow us to confirm those hypotheses and better predict the world around us. And it's an iterative process that enables us to refine our ideas over time. Those ideas are not set in stone. They are ideas that are changeable as we evolve or gain more information about the world around us, allowing us to more reliably predict the world around us and the processes that we're trying to study.
For more details on the scientific method, then I encourage you to go to my longer video just on the scientific method that you can find on my playlist on grapes and viticulture.
So let's review our plant anatomy. Plant anatomy is important for understanding plant function and physiological processes.
In this course, we will largely be addressing angiosperms, the flowering plants. In some cases, we may refer to the gymnosperms, which are primarily the pines and the fir type trees. We will not be addressing primitive plants in this course.
Plants have three major organs: roots, stems, and leaves. So the growth of annual plants occurs only at primary meristems. As you can see in this picture, there is a shoot meristem at the top of the plant where the shoot apex and apical meristem are shown. And there is also a root apical meristem, which is at the tip of the roots. At the base, you can see a root apex with an apical meristem. These meristems divide, the cells divide, and allow for the formation of new organs and the growth of the plant in a lengthwise manner. This image comes from the textbook Plant Physiology and was the textbook I used in my course, but I will be adding other figures of my own throughout the course as well. So you can see the three major organs here in this image. We have leaves, we have a stem that supports those leaves, and we have a root system composed of both in this case, a taproot and lateral roots. In addition, the taproots or roots can have root hairs in certain portions of the root. We will get into these details more in the future. The root apex also has a root cap, and between the leaves, we have what is called an internode, which is between the nodes. The nodes are portions of an axillary bud with a meristem that creates a lateral shoot to develop. So there's an additional meristem here for the beginnings of a new shoot, which again is at the tip, as in the overall shoot apex. At the top, the shoot apex, as you can see, both in the drawing and in the cross-section of a shoot tip, you can see the formation of what are called leaf primordia. These are the very beginnings of the formation of future leaves that will fully expand as seen in this image. In the root apex, in the image on the right, you can see the formation of this root tip and the root cap. This we will go into much more detail, particularly when we start talking about ion transport through the root. One final point is that plants that are perennials have secondary meristems which contribute to the diameter or the width of the plant, largely through the stems and the trunk. Now, within the angiosperms, there are two typical types of plants that we divide them into. We have the monocots and the dicots. The cots refer to their cotyledons, which are similar to leaves but not considered actual leaves. They form in the seed when the plant is developing. So in the monocots, they have a single cotyledon. With the dicots, they have two cotyledons. Now, they can be distinguished in other physical morphological characteristics as well. One of those is that the monocots, which are largely or composed mostly of grasses, but there are other plants such as palm trees, which are also monocots, they have a different form of leaf venation. That is, they have long, narrow leaves with parallel veins running down the length of the veins. In dicots, they typically have a broad leaf with a network of veins as seen in the image here. They also differ in their vascular bundles. We'll get into this later, but vascular bundles are pipes, systems in the plant that transport both water and sugars, and therefore, if you crop through a stem where this piping system exists, you'll see that the vascular bundles in a stem of a monocot are scattered throughout the stem. But in a dicot, they are more organized. As you cut through it, you'll see they form in the mature section of the stem a ring of vascular bundles. They also typically differ in their floral parts. Typically, a monocot has floral parts that are in the multiples of 3. And in dicots, they're in the multiples of 4 or 5. And also quite important is that they differ in their root system. So a monocot would have a fibrous root system, and a dicot would have a taproot system. We'll go into that again a little bit more, but basically you can see that a taproot is a major root that goes down with laterals branching off. Whereas with the fibrous root system, you have multiple roots growing out in a more fibrous, branched way. So as I mentioned, grasses are monocots, and they have a little bit of a different meristem. Rather than to have the apical meristem at the top, at the apical bud, as we see here on the left with a dicot, they have something called an intercalary meristem. This occurs at the base of the plant, just above the soil. Where the crown is listed there, there is an intercalary meristem at this base. So the leaf is actually dividing and growing at the base of the plant, as shown here in this figure. This is why when you mow a grass, it doesn't kill it, because you're just cutting off the top of the plant without damaging the meristem that is going to produce new leaves. Whereas if you were to cut a dicot off at the top, it would lose its apical dominance, as we'll talk about when we talk about the hormone auxin, and allow for the lateral branches to be unrepressed and start to grow, because that apical meristem is repressing those lateral meristems from growing. So in terms of floral components here, we can see on the left this reproductive flower that has modified leaves. That's what those petals are. And they have stamens and carpels. In the grass, they have an inflorescence. An inflorescence refers to multiple flowers. And in this case, they have spikelets and florets. I'm not expecting you to understand or memorize or know all the parts, but as a general rule, you should understand the different components of plant parts, as we will be discussing those in the future in terms of specific physiological processes. Let's look at the leaf organ. The leaf organ is made up of tissues. So tissues are made up of cells that have different types of cells in general. So in the leaf, we have multiple tissues here. We have something called the dermal tissue. The dermal tissue is the skin of the plant. So it consists of an upper epidermis on one side of the leaf and a lower epidermis on the bottom side of the leaf. On the bottom side here, it shows you a cuticle. This is an extra layer of wax that is put on the surface of the leaf to help prevent it from losing water. We also see different kinds of cells in this epidermal layer. So we have epidermal cells, but we also have guard cells. Guard cells are these round kidney-shaped cells that form around a pore called a stomate. This allows for gas exchange into and out of the leaf, as we'll talk about more when we talk about water transport and later in photosynthesis. In the center, we see the vascular bundle, which is consistent of vascular tissues, two tissues known as the xylem, which transports water, and the phloem, which transports sugar. We also have ground tissue, which is made up of cells known as the palisade parenchyma, or parenchyma, some people say, and the bundle sheath parenchyma, which are around the outside edges of the vascular bundle, encasing the xylem and the phloem. These parenchyma cells are involved in photosynthesis. And we have another name for that known as the mesophyll cells. The mesophyll cells consist of both those palisade parenchyma and the spongy mesophyll. The palisade parenchyma form a rather uniform layer just under the epidermis. The spongy mesophyll cells are more spaced apart. They're like a sponge in some ways, with spaces or holes in them that allow for transport of CO2 and water in and out of the leaf, because it's in an atmospheric transport process of vapor rather than in liquid form. Again, we'll talk more about this as we talk about water transport and photosynthesis. But suffice it to say, the leaf anatomy here is very important for its function. So when we look at the stem organ, we see it is also composed of multiple tissues, which are made of multiple cell types. We have the epidermis, or dermal tissue. We have the ground tissues, which are made up of the cortex and the pith, the cortex being just underneath the epidermis, and the pith being in the center of the stem. And then there are the vascular tissues, again composed of the xylem and the phloem. And note the location of where the xylem and the phloem are. The xylem are formed on the inner side, and the phloem is formed on the outer side of the vascular cambium. The vascular cambium is another type of meristem that gives rise to thickness or width in the stem. The vascular cambium gives rise to the xylem and the phloem. It is a meristem that also contributes to the width or girth of a stem. So it becomes very important in secondary growth, particularly for perennials, where they can increase their stem size over time. Particularly that of trees is most notable. Here we're looking at the root organ, and in particular, we're looking at a cross-section of a root in the mature zone of the root. It has an epidermis or dermal tissue. It has a cortex, which is part of the ground tissues. We have the formation of a pericycle or branch meristem in the center of the root. And this is protected by a layer just outside of it known as the endodermis. This will become quite important when we're talking about water and ion transport into the root. And in the central part of the root, also known as the stele, S-T-E-L-E, we have the phloem and the xylem tissues forming the vascular tissues, as we saw before. And there is a vascular cambium inside of here, which is also giving rise to the phloem, which is on the outer part of the stele, and the xylem, which forms on the inner part of the stele. Now remember, the xylem is responsible for transporting water, and the phloem is responsible for transporting sugars. In addition, in the epidermis, we can have root hairs. Root hairs grow out of the epidermal cells to increase the surface area, which improve water absorption in the plant and the increase in surface area for ion transport. But this only exists in a region just behind the growing part of the root, and as the root grows older, these root hairs disappear or are sloughed off as the root pushes through the soil. Okay, let's focus just on dermal tissues for a moment. They are made up of epidermal cells, as I mentioned. We also have ground tissue here, which are made up of parenchyma cells. So parenchyma cells are the basic form of cells that are formed in the meristems. And then later on, we have something called, after cell division, we have something called differentiation, where those cells may develop. They may not develop, they may stay parenchyma cells, but they may develop into other types of cells that we'll see in the next slide. So here we can see the differentiation of parenchyma cells into something called collenchyma cells. And these collenchyma cells have thickened cell walls that increase the strength and structure of those cells. We also have sclerenchyma cells, and these are even thicker cells, which are known often as fibers, adding strength particularly to the trunk. In the vascular tissue, as I mentioned, we have both the xylem and the phloem. And within the xylem, we have multiple cell types. We have cells that are known as tracheids, which are involved in the transport of water. However, in angiosperms, we have vessels, which are composed of vessel elements. And these have larger pores through the ends of the wall, and we'll get into this again when we get to water transport, which allow for more rapid water movement through a plant. So the gymnosperms and the angiosperms are differentiated in part by the fact that gymnosperms do not have vessels, but that angiosperms have both tracheids and vessel elements. In both cases, these cells are differentiated past the parenchyma cell and form what are called secondary walls, which are thickened walls of specialized function that allow for strength in the cell, but also movement of water. In the phloem, we also have unusual cell types that are formed that are known as sieve cells. And these sieve cells, again, are differentiated between gymnosperms and angiosperms. We find only sieve cells in gymnosperms, but we find sieve tube elements in addition in angiosperms that are not found in gymnosperms. And again, this has something to do with their organization and the movement of sugars through the plant. We also have structures that are in the sides of these vessels and tracheids and sieve cells that allow lateral transport from one cell next to the other that are built into these vascular bundles. In addition, there's a specialized cell in the phloem that is known as a companion cell. And when we get to sugar transport, we will speak much more about the interrelationship of companion cells with their sieve tube elements. So let's speak about a few basic parts of a plant cell. We have a cell that is consistent of a plasma membrane that surrounds the plasma inside the cell. So it's known as the plasma membrane. It is also the place at which the synthesis of the primary wall occurs to the outside of the cell, outside of the plasma membrane. So we call this area the apoplast, and we call the plasma membrane and the interior the symplast. So those are terms we're going to use throughout the course, and we will talk about symplastic pathways and apoplastic pathways. When two cells are attached, we have something called the middle lamella, which is a somewhat pectin waxy area in between the cells that helps to cement the two cells together. And we have some connections between cells that form a formation of a pore between the cells that we'll find out later on are regulated to some extent. Here it's listed as a simple pit, but they're also known as a plasmodesma or plasmodesmata for plural. And again, we will talk about their importance in transport between cells in a future lecture. Okay, so let's review some basic biology here. If we look at the parts of a cell, we can see If we start from the outside, the plasmodesmata that I was referring to here in the center going through the wall, we can see a plasma membrane forming around the cell, and again, the primary structures of the middle lamella, the primary cell wall, and the wall is largely made up of cellulose and some other materials such as pectins and hemicelluloses that, again, we'll talk about in a future lecture. In the interior of the cell, inside the plasma membrane, we have internal compartments known as organelles. The one that is very important is this one here called the chloroplast. The chloroplast is the site of photosynthesis, and there are many chloroplasts inside of a leaf cell. Chloroplasts are capable of dividing, and they have some of their own DNA. Another important energy organelle in the cell is the mitochondrion, or mitochondria for plural, as there are multiple mitochondria in a cell, providing the energy for the cell. We have other internal compartments, such as the Golgi body, which gives rise in part to other compartments, including the cell wall. We have the endoplasmic reticulum, which is another form of compartment, and we have what is known as the rough endoplasmic reticulum. They're known as rough because they have ribosomes attached to them, and ribosomes are responsible for protein synthesis, as we'll discuss in a moment. The smooth endoplasmic reticulum is a separate functioning compartment of the Golgi that gives rise to vesicles and the cell wall. Other compartments are known as the peroxisome. We'll go into their function later on. And we have a vacuole, which is shown here as a large blue bubble, and it's surrounded by a membrane known as the tonoplast. This vacuole used to be thought of as a garbage dump for the cell. We'll see it takes up a large volume of the cell, and a mature cell can up to 95% of the volume of the cell, with the remaining 5% being the cytoplasm, which is where all these other compartments are in. Another important organelle in the cell is the nucleus. The nucleus contains the chromatin and the nucleolus. It is surrounded by a nuclear envelope, which has pores in it that allows the transport of RNA from within the nucleus to the cytoplasm. The chromatin is a form of condensed DNA, and the nucleolus is a spherical structure that is the site for ribosome biosynthesis. So if we look at the membrane a little bit more carefully, we can see that the membrane is composed of certain components, and it forms what is called a lipid bilayer. So the lipids are a phospholipid. They have phosphate groups on the end. These phosphate groups are charged and more attractive to water, and the inner layers are lipid and repellent to water. So this naturally forms a bilayer where all of the phosphate groups are towards the exterior of the bilayer, and the lipid portions are in the interior of the layer. There are also proteins in this bilayer, and these proteins are often involved in transport processes, but they're also functioning as enzymes, doing biochemical processes that we'll talk about later on. So if we look at this portion of the phosphate groups, we would describe this region as the hydrophilic region, and we would refer to the lipid structure as the hydrophobic region, water-loving and water-fearing. In addition, some of these proteins have carbohydrate structures on them, and these structures are important both in function, but also in signaling and signal recognitions. Okay, I would be remiss if we didn't go through just a little bit about some of the basics of DNA, its structure, gene expression, and protein synthesis. So this is the most basic we'll get, and this is boring to you. You can move on, but this is very important for the functioning of cells and understanding the biology that we are learning about today. So DNA forms a double helix, which is then condensed and packaged around histones to form nucleosomes. And these nucleosomes continue their condensation process to form a chromatin fiber. As we look back further at these condensed formations of DNA, we can see there are looped domains and there are condensed chromatids which form the chromatids. So these chromatids are in a storage form of DNA, and this is unwrapped for the process of gene transcription or gene expression. So we have here the basic steps of gene expression. It starts with transcription. Transcription is the process in which DNA is read and expressed and transformed into an RNA transcript or message. These messages or messenger RNA are transported out through the nuclear envelope and transported into the cytoplasm. And this messenger RNA then is transported to the ribosome where protein synthesis occurs. Protein synthesis is the process of translation. It's translating that messenger RNA into a sequence of amino acids. And these amino acids are linked together to form a peptide. A peptide is simply a chain of amino acids of a certain length. And this then becomes, or can become, an enzyme, which is the machinery that is needed to process the message into some functional part of the plant cell. So we have ribosomes that are free in the cytoplasm, but we also have ribosomes that are attached to the rough endoplasmic reticulum. So both processes are occurring, and I am not going to go into the detail that is here in this figure regarding the actual steps of protein synthesis or translation. So transcription is the conversion of the message from DNA, so a gene or a sequence in the DNA that is transcribed into a messenger RNA. This message is then transported out of the nucleus into the cytoplasm, where it is translated into amino acid sequences to form the enzyme, the functional machinery of the plant cell. So in summary, plants are responsible for producing oxygen, capturing energy from the sun and transforming that light energy into carbohydrates for life on this planet. The scientific method is the basis of a reliable and predictive view of the environment and the world around us. Flowering plants are divided into two different morphological groups, the monocots and the dicots. And plant anatomy is important for understanding plant function and physiological processes.
Well, that's it. That's the end of this lecture. In the next lecture, we'll be talking about the importance of water. We'll talk about water properties, diffusion, and free energy and the water potential. This will help us understand why water is transported through the plant the way it is. After this, there'll be a quiz, so please partake in it and enjoy. It's not going to be difficult, but it will be a multiple choice test, and then it will be followed up with the answers in the following video after that. Have a great day.